So this video was involved in the National 5 Unit 1 part, which is called Cell Biology, and it'll specifically focus on genetic engineering. So genetic engineering is the 5th part of Unit 1, and it will cover essentially our use of other organisms. So it builds on the DNA information that you need to know from the previous few topics. So if you don't understand about what DNA is and what it's used for, you might want to have a wee look at that. So genetic engineering, we use it for to produce a lot of things, and what it involves is taking DNA and then transferring it from one organism to another. Now that DNA is in the form of genes. Now if you remember back to the DNA and production of proteins part of the course, genes code for proteins. So remember we talked about proteins being hormones or antibodies or structural, there's a whole bunch of other ones. So if you use genetic engineering, you can put one gene from one organism into another. And often what we'll use is we'll use a human gene and we'll place it into a bacterial cell. Now when we do that, what we call those bacterial cells is genetically altered, because we have changed the genes that's in them by then taking a human gene and placing it into them. Now there is a point to this, and that is that we can then produce useful human proteins from that. Now if you think back to the different types of proteins that we had, we talked about structural proteins and we talked about receptors and all that kind of stuff. Now the main ones that we then produce using genetic engineering are hormones. So if you think for a moment what hormones we talked about, one of them is involved in treating people with diabetes. So one main example and one we use genetic engineering for to produce is insulin. That's a really important one. Now, insulin is there to treat diabetics, so insulin will then help a diabetic to bring their blood sugar down because they don't produce their own insulin. So we can produce insulin using genetic engineering. We can also produce something called factor VIII. Now factor VIII is used for people that have what's called haemophilia. And factor VIII is this VIII is the Roman numerals for 8, so it's just the name of it. You could write factor VIII and it wouldn't really matter. So factor VIII is a blood clotting protein, so it helps blood to clot. So some people that are called haemophiliacs cannot clot their blood correctly. So what you would then use is a bacteria to produce factor VIII, and then we could give those people that can't clot their blood properly that factor VIII. The final example we talk about is human growth hormone. So I would imagine that most of you have heard of Messi, the footballer. Now, Messi is a very, very short man for just typical height. Now, Messi's actually shorter than he should have been, because Messi, when he was younger, was even shorter, and it was discovered that he didn't produce enough growth hormone. So what they actually did was they gave him human growth hormone and he grew more than what he should have done. So it was just replacing the deficit that he had. So people that don't produce enough human growth hormone, if they have a serious deficit or a serious deficiency of human growth hormone, they can then be given that to grow to a more normal height. Now, to get bacterial cells to produce our useful human proteins, what they have in them is called a plasmid, right, which is this structure here. Now you remember from way back in Unit 1, the very first part of Unit 1, we talked about plasmids being small circular pieces of bacterial DNA. Okay, so this is a bacterial cell. And this little circular piece of DNA here is a plasmid. Now this one here is a chromosome. You won't need to worry about that. Now notice it's a bacterial cell, it has a cell wall and it has a circular piece of DNA called a plasmid in it. Okay, so in the process of genetic engineering, you will need to know the stages and how this whole process works. So I've drawn a quick doodle, and what I've also done is written some stages out. So at the end I'll leave that on the screen and you can take a screenshot if you want. So this is a very rough diagram of genetic engineering, and it looks similar to the one that you have in your notes. So what you'll need to be able to do is label the structures that I've got on this diagram, and also be able to describe the process that's going on. So first of all, label this one. This one is a human cell. Now it's specifically, just generally an animal cell, but more specifically a human cell. Notice it doesn't have a cell wall. Okay, and this part here is the nucleus. Now we know that that nucleus contains genetic information in the form of DNA, which is then twisted up into chromosomes. Now this thing here, like I just labelled before, is a bacterial cell. And then that circular piece of DNA inside the bacterial cell is a plasmid. Okay, so what I'm going to do is I'm going to talk you through the stages of this. So the very first stage involves the human cell and getting the gene that we want out of that human cell. Now we do that by removing the chromosome that that gene is on. So basically this is what's happening here. So this is stage 1. Now that stage 1 is when the human chromosome is removed and then the desired gene, the one that we want, is then picked out on that chromosome. Okay, so like I've got in this description here, that human chromosome is removed from that human cell, and this is a chromosome here then. And this area I've highlighted here is the gene that we want. So this is our desired gene, the one that we would want to produce the protein for. So for example, that could be a gene that would produce insulin, or a gene that would produce human growth hormone, or a gene that would produce factor VIII. Now what we do next is we don't need the rest of this chromosome. So what we do is we then cut this gene out. So that desired gene, the one that we want, we would then cut that out, and we do that using enzymes. Okay, so the human chromosome is removed, that desired gene is picked out, and then that gene is cut out of the chromosome. Now then what we have to do is we go over to this bacterial cell now. Now this bacterial cell, if you look at this diagram, a part of that bacterial cell has been removed, and specifically that part is a plasmid. So that plasmid then is removed from the bacterial cell. Okay, literally you're just describing what happens here. This is a plasmid and it's been taken out of the cell that it was in, which is a bacterial cell. Okay, now the next stage below that then is that plasmid has been cut open. So it looks a bit like a horseshoe and it's been sliced open. And just the same as that gene that we cut out from the human cell and the human chromosome has been cut out using enzymes, so is this one. So that's stage 4, that plasmid is cut open. Now if you look at the next point here, this gene and this plasmid have then been combined together. So what's happened is we have then taken that desired gene and we have inserted it into that plasmid that we have cut open. So this here is stage 5. Okay, so next stage after that is we take this plasmid, which we now call our genetically altered plasmid, and we insert it into a new bacterial cell. So this is not the one that we previously had, because we've removed that plasmid and we've basically destroyed it as we've removed it. So we don't reinsert it, we put it into a new bacterial cell that was a host for it. Okay, so it is really important at this point in time to note that this is a genetically altered plasmid. It's not the same plasmid as we had before, because it has a new gene in it, that desired gene that we picked from the human gene. Okay, and it's inserted into a new bacterial cell. Now what you'll then notice is that this cell has then formed 2 cells. So that cell has grown and it has then divided, it's made a copy of itself. So that genetically altered bacterial cell, so again notice how I'm really emphasising the genetically altered bacterial cell has been changed. It didn't have this human gene before. It'll grow and then it'll multiply, it'll make copies of itself. And it'll produce that protein that's coded for by that gene that we put into it. So for example, if we had taken the insulin gene and put it into that plasmid, this cell here would produce insulin. If this gene was a human growth hormone gene, then this cell here would produce human growth hormone. If this was factor VIII, then this cell would produce the factor VIII protein. Okay, so it produces the protein that we want. Now this is not always insulin. Okay, this can be another example, like I've listed. Okay, and then what we can then do is we can extract the insulin or the other protein that this cell produces, and we can purify it and then give it to the people that would need it. Okay, now one more wee point to note is just the enzymes are used in this process. So specifically, enzymes are used to cut open or seal in pieces of DNA. Okay, so I'm going to leave this for a wee second in case you want to screenshot it. And then here's your description of those stages. Okay, and that gives you a run through of the whole process. Now I'll post a quick scan of these diagrams up onto the Google Classroom just so that you can see them both separately. But that runs through the whole process of genetic engineering.